All right. So thank you everyone for joining us. This is the Black and Micro Week Microbiome panel. I'm really excited about the conversation we're going to have today. My name is R.A. Kozik. I'm a postdoc at the University of Michigan. I study the respiratory system microbiome and how it interacts with asthma and how it may be involved with asthma phenotypes. I want to thank each of our viewers for joining us, and a special thank you to the panelists for coming and being part of our conversation. First, I'd like to acknowledge the sponsors of Black and Microbiome, or Black and Microbiology Week. We would like to say a special thank you to our gold level sponsors, which include the American Society for Microbiology, the American Society for Virology, and QIAGEN. Their generous support is instrumental to the success of our programming this week and beyond, and we appreciate their continued efforts in supporting and recognizing Black microbiologists and their contributions to science. So today we're going to talk about microbiomes. Microbiome is a community of different microorganisms living together in a particular habitat. So all humans, animals, plants, and environments possess unique microbiomes. We know that these communities are critical to the stability of our world. For example, the ocean microbiome is responsible for generating over half of the oxygen on Earth that we breathe, and soil microbiomes are very important for the global processing of nitrogen cycling and food production. And of course, we also know that, or we're learning that the human microbiome is very important for maintaining human health. So our wonderful panel today consists of researchers who study the human microbiome. We have with us Dr. Leah Guthrie, Dr. John Brooks, Dr. David Pride, and Dr. Candace Price. So first, for our first question, I'm going to ask each of the panelists to introduce themselves and kind of give us and tell us about how they came to be studying the human microbiome and a little bit about their work. So Dr. Brooks, if you'd like to go first.
Sure. My name is John Brooks, and I too am from Michigan. I went to the University of Michigan for my undergraduate work. After undergrad, I went to Northwestern University in Chicago, where I studied with Mark Mandel, and I pursued questions concerning a symbiotic relationship between the Hawaiian bobtail squid and its bacterial partner Vibrio fischeri. This was a model microbiota organism in terms of trying to identify the mechanisms in which an animal host acquires a microbiota from its environment. After that, I went to the University of Texas Southwestern, and I'm currently studying with Laura Hooper, and I'm a Hannah Gray Fellow here. And I'm interested in understanding now more how the bacteria that reside in the gut regulate host immunity across the day-night cycle. We've come to know that the microbiota is a huge regulator of host immune systems and host susceptibility to infection. And I'm kind of adding a new dynamic to this in terms of the day-night cycle being a regulator of this as well. So.
Let's have Dr. Price go next.
Hi, so I received my PhD in endocrinology, so not in microbiology. And my focus has always been understanding type 2 diabetes prevention, obesity, and cardiovascular disease. And so my work has mostly focused on understanding insulin resistance development and the mechanisms behind that. I started off looking at adipocyte function and doing a lot of animal work, moving into more human work, which is what I'm doing now. And when I went to my first postdoc at Stanford, one of the projects that we were collaborating on with another investigator was looking at the microbiome in a longitudinal study to understand the link between microbiome and insulin resistance development and potentially type 2 diabetes development if anyone in the study did become type 2 diabetic over the course of the study. That was my first introduction really into the role of microbiome and health. And that really sparked my interest in pursuing this area of study. It's not my complete focus, but it is something that I now incorporate into my studies, looking at the gut microbiome and how that interplays with nutrition. So my focus, my overall research focus is looking at the metabolic effects of sugar sweetened beverage consumption and how that increases our risk for cardiometabolic disease. with a particular interest in looking at Black women. And so now I am incorporating the use of microbiome technologies to understand if there's an interaction with high sugar intake in the gut microbiome and how the gut microbiome may play in the development of insulin resistance.
Excellent. So interesting. How about Dr. Pride?
Hi. First, I'd like to start by saying thank you. It's really a pleasure to be here to talk to everyone really about what we can do and about what we're doing. And I'm happy to, if anyone wants to reach out to me. I will say that sadly, I tend to be a little busy nowadays during COVID times, but I'm happy to try my best to get back to anyone because I am very interested in inspiring sort of the next generation of minds out there. So I am David Pride. I am a researcher as well as a clinician. I am originally from Nashville, Tennessee, and I sort of have trained all over the US. I do have something in common with Candace as we both ended up doing a postdoc at Stanford. I honestly have just been interested in microbiology most of my life. And, we focus more specifically on the viruses that are part of the human microbiome rather than the bacteria. But we also do some more sort of I hate to use the word generic bacterial microbiome studies in my research laboratory as well. Now I'm out at the University of California, San Diego, where I'm an associate professor. And the one thing I will say is that I don't know if people have seen me before, but one of the things that's probably a little interesting about me is that when I was originally applying for faculty positions, I was applying during the sort of economic meltdown of 2009. So I ended up taking a position that is really a bit odd, maybe, and maybe different than what everyone else is doing. Is I took a position as the associate director of microbiology. So I'm running a clinical microbiology laboratory at UC San Diego. So right now I'm also at the forefront of all of the COVID testing at my university. So it's been a really busy time, but COVID is microbiology as well. So even though my heart is in the human microbiome, anything microbiology related is something that I'm just really very interested in.
Great, thank you. Dr. Guthrie?
So hi, I'd also like to say thank you to the Black in Microbiology team and for inviting me. It's been great to see this take place so far. So currently I am a postdoc at Stanford in the Department of Microbiology and Immunology in Justin Sonnenberg's lab. And I study how gut microbes process foods and drugs and the consequences of this metabolism for human health. And in terms of how I got into microbiology, I would say early on in grad school, what kind of led me to study the human gut microbiome in particular was just the appeal of studying a complex microbial community. And for me, I was really interested in learning about how we can understand how you go from very many small localized interactions between specific species or specific metabolites, and how that results in these kind of emergent phenotypes and for human health, whether it's changes in human nutritional status or how we metabolize drugs. And so that's really kind of the introduction, is that I like studying things that are kind of these complex biological systems, and the microbiome is really a great place to be if that's what you're interested in.
Great, thank you all. So my next question for you is thinking about the complexity of microbiome research, because we are sometimes maybe focused on bacteria or viruses, but having to keep in mind that these organisms that we study are in community and not isolated. So given that there are experimental design and statistical considerations that we have to think a little bit more about, because we are looking at this from a more of a complex system. So because of that, there's a lot of times where we have to borrow from other fields or collaborate with other fields to kind of get tools and techniques to answer the questions that we're interested in. So I want to know, have any of these collaborations where you're kind of going outside of microbiology been a part of your research? And what role does that play in the way that you investigate the microbiome?
So I actually will sort of address the opposite of that, because I'm not in the field of microbiology, and that's not where my training is. I actually collaborate with microbiologists to help me investigate the microbiome. And so I'm a faculty member at UC Davis now, and we have a microbiome core there. And so through that core, I'm able to collaborate with experts in processing these fecal samples that we collect, but also in all the bioinformatics that needs to be performed in order to analyze the data. That is not my strong suit at all, especially the statistical programs that are needed to do these analyses. That's not where my training is. And so I've become over time familiar with understanding how the data is analyzed, but because I don't have that specific training, I always collaborate with other people that do. And so, that's really how I like to conduct my research in general. I think to solve big health problems, you really need to be interdisciplinary and collaborative, and, expand your research questions by working with people that are outside of your field. So I definitely do that a lot in my work.
Yeah, just to add on to that, I would say that when you study the human microbiome, I'm focusing on the gut just because that's what I think about mainly. It's an inherently interdisciplinary science, and just kind of getting at your question of like, well, what do we, how we kind of address challenges in terms of the statistical or experimental challenges. I would say that I guess I'll just highlight a few things. I think in terms of how I study the microbiome, I'm looking particularly at metabolism. And so we do a lot in terms of metabolomics and trying to understand, well, what are the molecules produced by the gut? And so with that, you need kind of like expertise in studying and looking at and quantifying metabolites. And then also kind of our first window, and I think why so many people got excited about the microbiome was just kind of advances in how we can sequence the microbiome from a stool sample in a non-invasive way. And so with that, it really requires an understanding of dealing with the complexities of kind of the sequencing kind of process and the errors inherent in that, as well as kind of building new bioinformatic methods to deal with those approaches. And so I would say, I think it's a field of study where I think a lot of different people from various backgrounds can have a lot to contribute.
Sure, and I would add my bit of a story about some of the things that we've done in the human microbiome. So I came into microbiome research in a bit of an interesting way, in that my background is actually in computer programming. And when microbiome research started to become much more popular, really about 15 years ago, it was very difficult to do. Part of the reason it was so difficult to do is that there wasn't the same technology then that we have now to be able to do it. But a lot of that technology was also on the informatic end. So my sort of introduction to human microbiome research was that there just wasn't any way to interpret your data. You could sequence it, but you couldn't interpret it. So one of the things that I started doing, particularly for the human virome, is just writing software to be able to analyze all of the data that we were generating. The nice thing about microbiome research now is that you don't really have to write your own software anymore. It's just very, very easy to go out there and find somebody else's tools, mostly tools from people who are much better programmers really than I ever was. But it's really nice to see how this field of the microbiome has shifted so much over the past 15 years, where now a lot of this stuff is a lot more intuitive, a lot easier, and it's open really to people. I mean, you don't necessarily have to have a background where you started out being taught in the human microbiome to be able to get into microbiome research, because there are a lot of tools and a lot of people who are cross-disciplinary that you can collaborate with nowadays, so that it's basically sort of democratized where just about anyone can do it if they're really interested enough.
Yes, so in my day-to-day, or at least within my project, I was trained as a bacterial geneticist, but the questions that I've begun to ask center more squarely on immune system and immunology. And so I've had to partner with immunologists in the lab to kind of learn those skills and techniques, just to be able to address the questions that I'm interested in addressing. But I would echo something that was said earlier, that studying the microbiome is inherently interdisciplinary. There's just so many aspects of study that require you to have multiple skill sets available to you. And so that's definitely, collaboration has been an underpinning of some of the work that I've done. But even beyond just the general day-to-day, I've also engaged in collaborations with another lab. So I've become interested in questions of how viruses interact with the microbiota and how that impacts host immunity. And so I've done some collaborative work with Michael Acevedo and Julie Pfeiffer's group to kind of get at these questions in a different way. So I think collaborations just in general are going to allow us to really pin down some of these really general, more difficult to answer questions.
Great, great. Yeah, it's been my experience also that collaboration has been really important in thinking and looking at data and figuring out how to go about addressing new questions that may arise. So it's definitely an important facet of the field, I think. So thinking this could either be about your particular research or the field of microbiome research more broadly. What recent development or possibility or potential application of microbiome research has you the most excited these days? It seems like the field moves really quickly. So I'd love to hear about what potential applications are you most excited about?
Well, I'd be happy to give a bit of a generic answer to your question. You know, one of the things that, my lab is really interested in is the role that viruses play in the human microbiome. And I'll sort of put a bit of a teaser out there, but, one of the sort of sentinel studies in microbiome research was Jeff Gordon's work, which, goodness, may be 20 years old now, where those guys took a group of mice, fed them high-fat diets, and of course the mice got fat, right? And then they took a group of lean mice, fed them lean diets, and the mice stayed lean. But then what they did was they did a transplant of the microbiota from the gut of one set of mice and put them into into the lean mice. So in other words, they took microbiota from the obese mice, put them into the lean mice, fed them lean diets, and of course the mice got fat. And that was one of those sentinel studies that sort of taught us that, your microbiome can affect your metabolism so significantly. One of the things that we've been doing is looking at the role of viruses in there, because most people have just made the basic assumption that, oh, well, the mice are getting fat because the bacteria in their microbiome are doing metabolic things that are making them fat. The one thing that most people have not paid attention to is the fact that they were also transferring viruses. So we've been looking at what the role of those viruses are that are being transferred. And the one thing we can say is that you can take just the viruses and reproduce that phenotype, meaning that if you take viruses from the gut, those mice will get fat. And of course, the question is why. We don't know why, but those are the sorts of things that we're really working on. And it's, I think it's why sort of the viruses in the microbiome are so interesting to us.
Dr. Pride, when I first became introduced to the microbiome, the field of microbiome, I read that paper that you referred to, the Gordon paper, I think, and I was discussing it with a resident that I was an acquaintance, and he didn't believe that this was true data, that the microbiome was really important, and it was shocking to me. But anyway, I'm glad that the field has blown up, and clearly there's consensus that there's, an important role for the microbiome. For me, I'm particularly interested and excited about the potential of developing personalized nutrition interventions in people based on their microbiome profiles and how their microbiome may interact or respond to certain foods and nutrients and micronutrients, and being able to tailor specific diets for certain people that may be at risk for metabolic disease, or maybe are obese and need a better diet regimen that works well for their body, because we know that not everybody has the same microbiome profile and not everybody may respond to the same dietary intake. And so that's what I'm interested in the most. I know some companies have started, there's some startups, I think, trying to get into that area already. I don't know if it's too premature or not, but that's what I'm excited about the most.
Great. Anyone else?
Oh, I was just gonna add that I think in the beginning of the microbiome, there was a lot of interest in 16S sequencing and then correlating microbiota composition with X disease state. And I think we're getting to the point where we can get a bit more mechanistic in determining which functional groups or which bacterial groups are responsible for some of the phenotypes that they've been correlated with. And I'm looking forward to the point at which we can just edit the microbiome and get rid of specific bacterial groups or specific bacteria that produce X metabolite, and then ask more mechanistic questions about what that does and what impact that has on human health and fitness.
Yeah, I guess I would also piggyback off of that. I think so as a part of my work, it involves kind of using dietary intervention studies to get an understanding of how the relationship between diet and the gut microbiome and human biology. And I think what I'm really excited about is that I see these studies, I guess, so there's the technical aspect that that's exciting in terms of learning like how to study the microbiome. And I think that what we're learning is that your community composition can tell you one particular thing about your microbiome, the metagenomic, or so basically the collection of genes, and kind of investigating that by doing, say, shotgun metagenomic sequencing may tell you something else about the microbiome. Profiling metabolites may tell you something different about the microbiome, as well as profiling proteins. And so one thing that I'm excited about is just the way in which kind of the tools and the ways that we can probe the microbiome are expanding, as well as the way that we're thinking in terms of the theory about that we have. And so kind of like John was saying, I guess kind of, I think we went through the kind of this stage of really looking at like, wow, like in different conditions, you have different community compositions based on 16S of the microbiome. And so what does that mean? And I think kind of this word came up called like dysbiosis. Oh, well, you have a dysbiotic gut. And I think that what we're moving from is from kind of these more nebulous terms to really, again, still using ecological principles, but in more targeted ways to understand the differences between the ecology of the gut versus like a rainforest or some of these communities in which we're pulling some of these principles from. And so I would say that there are technical and theoretical developments. I think people are really getting better at thinking about how to use these tools to get at mechanistic insight. And then in terms of the insights, I'm also really interested in kind of the role of diet and the gut microbiome. And for me, I'm really excited about prospects of being able to control an individual's metabolic output to either increase or decrease the abundance of particular metabolites that we think are helpful or beneficial in a disease context.
Yeah, that's really a really interesting and important point to remember. I often think about how we know that there's really high variation from person to person in the microbiome. And that's like one of the challenges we have, because I know in animal work, like we kind of depend on the fact that those microbiomes are going to be like replicated so that we can use the math that we have to figure out what is important. So that was a good point about being able to think more ecologically and how to apply those things to humans specifically, especially in the context of everyone's microbiomes being inherently different, and how do we like use that and like where can we generalize and where can we maybe not generalize? And maybe we should be paying attention to where we can't generalize because we might find some answers there. So I have one more question and then we're going to open it up to the attendees. So if you are watching and you have some burning questions, feel free to submit it through the Q&A and I will present it to the panelists. So my next question is, in your view, how or what about microbiome research particularly impacts the Black community? As we know, we often have disproportionate burden of a number of health conditions for a variety of reasons. And currently it's common to just kind of classify people based on their background and look at their microbiome and say, oh, this is like what's different. And so maybe this has something to do with the phenotype or the symptoms that they're experiencing. So I guess I'd like to hear how each of you think about microbiome research in the context of the Black community.
So for me, it's maybe two thoughts. One, obviously the role of the diet and how that interacts with the gut bacteria that is maybe most predominant in certain populations and certain ethnic groups or race groups, but also from an evolutionary standpoint, what is the role of the gut bacteria when you have now taken a large population away from their typical traditional culturally relevant foods, and now you've placed them into a more westernized tradition where now they're getting completely absent exposure to their traditional foods, and now that gut bacteria that was essentially developed to interact with those foods that they were eating and maintain that healthy metabolism and health, and now you're exposing them to different foods that bacteria that existed before isn't used to processing. And so how does that now play into increasing our risk for obesity and metabolic disease? Is that changing how we respond to certain foods? Is it also forcing us to maybe reconsider getting back to more traditional foods that our ancestors are, used to eat? And is that something that our body prefers, eating more vegetables and fruits and things? But just thoughts like that is something that I'm interested in, looking at different groups that have been exposed to this nutrition transition, which is basically when your nutritional environment changes from your traditional foods and into something completely different, and in most cases it's a more westernized diet. So I think that could play a role and explain some of the mechanisms that link poor nutrition in particular, to obesity and cardiometabolic disease, particularly in the Black population.
That's really fascinating. I like the concept of nutrition transitions. I don't think I've ever heard it articulated that way, but I almost wonder if that is something that is important even in the short-term scale. I mean, if you're growing up all your life and someone is providing meals for you, and then you like move out on your own, and you kind of go through a transition then where you decide like how you are going to like what your diet will be like as you grow older. And I wonder like what impact that would have. I mean, surely your microbes would have to adjust. So that's a really interesting thing to think about. Anyone else have anything to add?
Yeah, one of the things, and I hate to sort of harp on the diet thing, but the way I think a lot of us think about the microbiome is that you sort of are what you eat. And there have been studies that show, hey, you can take someone who's, for example, not eating a westernized diet, and you can put them right smack dab in the middle of the US, and their microbiome will transition fairly quickly to adjust to the changes that they have in their diet. And that does have impacts based on whatever the traditional foods are within whatever community you're looking at. So that if your community is eating a lot of, sugary foods, et cetera, you will see that their gut microbiomes, for example, will change to reflect, in essence, what they're eating. So when we look at the microbiome, one of the big concepts that's really very interesting to us is the concept of sharing, meaning that we not only think about the microbiome in the context of, I am my microbes, but we think about them in the context of not only am I my microbes, but the people who are around me and touching me and, sharing the same resources that I am every day are also sharing our microbes. So when you get communities that to some extent are different from others, or aren't necessarily interacting with other communities, you will start to see this sort of drift in the microbiome. And we can evaluate that when we look at different races and ethnicities. You can start to see differences. Maybe they're based in diet, maybe they're based on just being around people who share. But in fact, we do see those sorts of microbiome differences that we can trace to individual populations. And one of the things that we're really very interested in is, well, what is the effect of those different microbes? I think the way a lot of people look at it is, well, you might see one set of microbes, but they're actually doing the exact same functions that this other set of microbes in a different population is doing. So we're not yet at the point where we can say this microbe is good or this microbe is bad. But I think that's where a lot of us see sort of the future of the microbiome research. How do we manipulate the microbiome so as to promote health? And we'd like to be able to do that really in all populations and not just base it on, the majority population.
Yeah, I'd like to add a few things. I guess one thing that I would say is that there have been a number of studies that kind of show, and so looking at microbiomes of individuals from people in different places, North America, South America, various places, you kind of see that as for some communities, as they kind of migrate from, for example, one country to say the US, you see by like the second generation, their microbial community kind of shifts to a more generic North American microbial composition. I guess one thing, and so my lab, in the lab that I'm in, one of the key things that we study a lot is just the idea of the role of fiber and the importance of fiber and kind of feeding your gut microbes and what that means when you have low fiber diets, as we do in the United States, for our health and the ramifications of that. And so microbes are really important for breaking down a lot of these complex carbohydrates that our human enzymes can't into products that are really vital for our health, such as short chain fatty acids. And so one idea, and I won't get too much into the weeds, is that when we don't supply it enough of these complex carbohydrates, gut microbes instead look for what's around. And what's one type of food that's around is kind of this thing called mucin, which is in your gut epithelia. And so it's like kind of eating at you instead of eating these fibers because you're not supplying it. And so that's kind of one thing. Again, I think diet is really critical across all populations and kind of thinking about, well, what microbes and what functions are we missing because we're not applying, giving our microbes the right foods and substrates that it needs to produce these metabolites that are really important for our health. I would also, just one more thing, it's kind of more of a general science thing and not just a microbiome thing. I think that, so one of the things is I think we've all seen kind of like a lot of companies kind of get into like trying to use the microbiome for various reasons, whether it's like they're making their own probiotics or like acne treatment or whatever it is. And I think that, or like kind of, I remember kind of papers that kind of look at, well, like, okay, let's kind of make a predictive index of like what makes a healthy microbiome. And I think these are all really valuable things. But I think that whenever we do this, just as like when you're kind of the general process of drug development, you kind of have to study a diverse group of people. It's not necessarily because there's something inherently biologically unique about Black people. It's just the idea is that diverse groups of people have diverse experiences that are going to impact how they respond to things. And so that I think it's really important for microbiomes and people who use the microbiome studies to not say like, oh, well, I found these 20 people and so I'm just going to study them. And yes, they're not really diverse, but we'll do that later. We didn't have time. And I think we really have to think about who we study when we're in like what it means for our conclusions. And so, and then like one last thing that's kind of very important to me in terms of like the idea of like, well, what is the unit of microbiome that we're trying to target and change? And so for some people, I think they would say like, oh, we're trying to, I think it started off really if we're trying to change specific microbes. And I would say that for me, kind of how I look at it is that you will find different communities. For me, it's kind of like, well, there are different communities or like small communities called guilds or groups of microbes that may be able, like you produce a lot of this compound and then you can, another collection of microbes produces a little bit of it. And so in different people, the set of microbes may be a bit different, but I think we're learning that it's not necessarily going to be the one species across everyone that does the same thing, but you're going to have community members that are really good at doing certain things. And we're trying to figure out, well, what is that complete guild of community versus like that one species? That's, yeah.
Right, those are all really great. Thank you for those responses. So
Oh,
now,
I was just gonna quickly add, I'm so sorry.
go ahead, no problem. Go ahead.
Just in terms of how the microbiome impacts specifically the Black community, I think it's been shown that the greatest driver of microbiome composition is diet, and that genetics has very little impact on the overall changes in the microbiota. So it would be more of like, in my thoughts, a general impact of how diet impacts microbiota composition and how that impacts host health and fitness, as opposed to a question of host genetics. So I guess I just wanted to add that I don't know if there would be a specific role for the microbiome in terms of the African American or Black community. Yeah.
Right, great. I think I would agree with you there, because I think a lot of times we tend to think, well, you have different outcomes in different groups, so there must be something inherent in the groups that is causing that difference. But I'm encouraged to see that we're slowly starting to think a little bit more outside the box and figure out what else is going on that might be peripherally related that's actually at the root of those differences. So yeah, so now I'm going to ask some of the audience questions. So some of these are a little bit targeted to different ones of different research that you guys engage in. So just feel free, if the question speaks to you, to jump in to answer it. So since we've kind of been on talking about diet a little bit, someone asks, what are your thoughts on probiotics? Are probiotic foods as important as we are told, or is it just a fad?
Well, I'll certainly give my 2 cents about probiotics. And that is, the one thing that I often teach when we talk about probiotics is, not every probiotic can work in every person. And part of that is just what is the microbe that you're putting in. So if you're someone that's taking probiotics, you might really want to think really hard about what is this microbe, and is this microbe capable of growing? Is it capable of competing with my own gut microbiota? Because a lot of times we see people who are taking probiotics with organisms that can't even grow in humans. And while it's possible it could have some effect, even if it can't grow, you're certainly limiting yourself significantly. And I'll sort of add an anecdote from a study that we did but never published. But one of my colleagues, who's very interested in his own personal gut microbiology, who collected his stool every day for the course of a couple of years, which is, that's pretty extreme. But we also did the extreme thing of sequencing his gut microbiota every day for a couple of years. And during that time, he had been on several different over-the-counter probiotics. And the one thing I can say is that from deeply sequencing all of those specimens, we never once found that probiotic in his gut microbiota. So, if you're going to use probiotics, the one thing to really think about is don't just believe what the label tells you, but do a little bit of homework background to understand, is this something that might actually be capable of working in me? Because so many people just throw so much money away on probiotics that aren't really capable of doing anything for them.
Yeah, and I'd just like to add to that. And so, so I guess on one hand, it may be the case that the probiotic that you're taking, the gut microbe, may not need to colonize your gut to be efficient. Maybe there's a protein or bacterial protein or something that's important. On the other hand, I would say that for what the research shows, that your habitual diet is what really is kind of the biggest component. And I think, and so, I guess kind of to say this briefly is I think there have been studies that showed that so if you have again kind of like a low fiber, just kind of like if you have an habitual diet that isn't really feeding your microbes, I guess the impact of a temporary or like taking a probiotic isn't really going to overcome or surpass the challenges of having a diet that isn't providing the substrates that your gut microbiome really needs. And I think that's what's really interesting about a lot of these dietary intervention studies is that for in terms of who responds or doesn't respond to a probiotic, I guess it really, again, it's about the diet that they had going in, and like it's kind of the makeup of your microbiome that you had before you started. And depending on your makeup, you may be more or less receptive. And so I think there are a lot of factors. And so I feel like the only thing that I would say confidently is like eat fruits and vegetables, eat a good diet, and we'll see what the future holds for probiotics.
All right, great. We're going to pivot a little bit. So the next question is, what do you think of the potential bacterial viral crossplay or crosstalk in respiratory infections, for example, SARS-CoV-2 or influenza? Should we be putting more emphasis on the more complex interactions within the microbiome?
Yeah, I wish I had a great answer for that, to be honest with you. The one thing I will say in this, I don't know what people are teaching nowadays, but it's important because it's much different than what I was taught. When I was, taught specifically about viruses, the way we were taught is that you get one viral infection at a time, and that those viruses tend to compete with one another so that you almost never will get multiple viral infections at the same time. And I think one of the things that we've learned, and a large part of that, is that now we have more sophisticated testing for individuals who show up with respiratory illnesses, et cetera. We see people all the time that show up with influenza and rhinovirus, for example. At the beginning of this pandemic, we saw quite a few people showing up with both influenza and SARS-CoV-2. So it's absolutely possible to be dually infected at the same time. What that means for the people, unfortunately, we don't fully know just yet, as I'm not certain that the research is there, but I think now that we know that people can have dual infections, and that we need to be trying to diagnose these dual infections at the same time, that helps us understand that we probably do need to study the consequences of having these dual infections. So I think probably everyone would be for studying these things, but unfortunately SARS-CoV-2 is just so kind of new that these sorts of studies might be years in the making rather than months in the making.
Great. All right. Our next question is for Dr. Brooks specifically. How does the microbiome change with the circadian rhythm?
So there's been a huge interest now in the role of environmental light cycles and directing rhythms, not just in our own biology when we eat and sleep, but also what impact this has on the gut microbiome. What we know is that one of the greatest drivers, again, in the microbiome composition is feeding, your diet, but also feeding behavior. So throughout the day-night cycle, when you eat has an impact on the abundance of the microbiota and the composition of the microbiota in the gut. And a lot of my work is centered around determining how these diurnal changes in the microbiota impact host health and fitness, or how it regulates host immunity. And what I'm beginning to show and beginning to learn, and there's some work out there right now that indicates this, that eating and feeding has an impact on the overall composition of the gut microbiota, and this can dictate your susceptibility to infection. So at least for salmonella, you're actually more likely to get a salmonella infection or a productive infection when you eat as opposed to when you're not eating. Coincidentally, the likelihood of you getting salmonella, which is a foodborne pathogen, occurs when you eat. So it's an interesting dynamic between resistance mechanisms of the gut microbiota and pathogen infectivity. And just as an aside, for the previous question, it was about virology and impact on the microbiome. I can't speak specifically to respiratory viruses, but Julie Pfeiffer's group has shown that for enteric viruses, they actually interact with members of the microbiota and they stabilize the viron, and it allows you to get enhanced infectivity. So enteric viruses actually depend on, in this trans-kingdom interaction, members of the gut microbiota to infect host cells. So there is some cooperation there that her group is really interested in discerning more. But yeah.
Great. The next question is, how much do you think that the human microbiome influences mental health or mental illness?
Well, yeah, I would say that's a really interesting question. And folks have really started to study that, particularly from the perspective of autism. And it's really very interesting that you can find reproducible differences, for example, in your gut microbiota in autistic patients versus non-autistic patients. And part of the interest is, well, is it a result of the disease that the gut microbiota are different, or are the gut microbiota being different sort of more of the cause or what is exacerbating the disease? And, folks have been working in murine models of autism, where the beauty of those is that you can control the microbiota in some of those. And when you put certain groups of microbiomes, or you basically prevent certain microbes from colonizing the gut, you can actually reproduce some of those. I forget the term of the behaviors they call them in autism, but you can reproduce some of the autism-like phenotypes in mice just by manipulating the gut microbiota. And that helps us understand this thing that is sort of out there, of this idea of this gut-brain axis, where the microbes and the metabolites that they're producing actually are, in essence, communicating with all of the nerves that you have in your gut, and that information is being communicated back to the brain. So absolutely, I think most people at this point will say that your gut microbiota can have significant interactions with the brain and can have some significant role in determining your behaviors moving forward. So I see that as sort of on the vast horizon of where so much important microbiome research is going to go. And, it makes it all the more important for us to understand how we can manipulate the gut microbiota so as to promote human health, because there might be a lot of things, including these sort of anxiety-like behaviors and autism, that you can have some measure of control over just by manipulating the gut microbiota.
So Elaine Hassal's group at UCLA has shown that there are definitely bacteria in the gut that are making neurotransmitters. I think as of yet, it's unclear whether these neurotransmitters that are being made in the gut actually cross the blood-brain barrier and impact brain activity, but there are a series or a huge population of enteric neurons that line the gut, and there is cross-communication with those. But I think opening the door and trying to understand this gut-brain axis that David was talking about is definitely the next frontier in terms of how neurotransmitters made in the gut impact host health, and how a lot of the metabolites that are made by members of the microbiota impact host behaviors.
Great, great. Yes, that is fascinating. We have time for a couple more questions. So first, I'd like to hear from each of you what topics or microbiome subfields do you think are understudied or underfunded or need more consideration? So if you had the purse strings, as it were, where would that funding be going? And we'll start with Dr. Price.
Okay, well, I'm a little biased because it's from personal experience, but I'd say more funding towards microbiome research geared towards understanding how specific diets are impacting metabolic health. There is certainly some microbiome funding for nutrition-focused research, but not just a personal anecdote. We've had reviewers basically tell us to take out the microbiome portion of our grant that was focused on understanding sugar sweetened beverages and its impact on cardiovascular risk factors. And so they didn't want to fund that part of the grant, but they liked everything else that was mechanistic, focused on looking at biomarkers of cardiometabolic disease. So I would like to see more work in that. And then also, there's very little literature on the role of microbiome in health disparities and specific in various ethnic groups that are where there's a lot of health disparities, particularly cardiometabolic disease. So I'd like to see more funding and work in that area.
Dr. Guthrie?
And so in terms of what is understudied, I guess, so in terms of areas that I'd like to see, that I do see, but I'm looking forward to see more studies, I would say that, I guess when you think of what we've been talking about, a lot of the impacts, whether it's on mental health or cardiometabolic disease, it's really kind of the unit of action are these microbial metabolites that are they're interacting with a host receptor and leading to this activating some host pathway. Or in the case of what I used to study in grad school, they're reactivating drugs that we take and causing adverse reactions. And so I think that it's really important at the tool level. And so the sequencing tools that we have are really amazing. But I think we also need to kind of expand our understanding about the enzymatic and chemistry of microbiome. And so I think that's a lot. There's enzymology, there's chemistry to be done to really expand what we can do. And so like for all of us, so for me, when I'm analyzing kind of human microbiome sequencing data, there are many unmapped reads, right? There are many unknown genes. And I think we really need to start assigning functions to more of these things. Conversely, I think we also, and I think this is a growing process, but really understanding the pool of microbial metabolites that we haven't yet really characterized. And so I think studying enzymes and studying metabolites at greater detail is really important.
That's an excellent point. I know sometimes you get metabolites and they're like XYB 21357. I'm like, oh, that's great, but I don't know what that is or what it does. So yeah, I think developing the tools and technology will really help move things along. Dr. Brooks?
I'm going to piggyback off of Leah. I think drug metabolism by the microbiota is probably really the next frontier. I mean, there have been individuals who have shown that there's a lack of drug efficacy for some cancer drugs and that this is due to microbiota composition. I think kind of underpinning mechanistically what's going on there could improve overall cancer treatment across the board. So I think that that's definitely an area that needs much more funding and investment, for sure.
I definitely agree with that. We are looking at that in asthma right now because there are some patients who don't respond well to inhaled steroids or other therapies, and we're seeing a signal that their respiratory microbiomes are a little different depending on whether or not they're responding or not. So that's really, really important. And Dr. Pride?
Well, I would sort of put in a plug for viruses, of course. I think you're kind of getting all of our interest as what we think should be the next thing on the horizon. But one of the things that maybe most people here don't know is that your body is just full of viruses. You've got, 31 trillion viruses in your body. Many of those viruses are bacteriophages, which means viruses that just infect the bacteria. And one of the things that we've really been moving towards is this idea that antibiotic resistance has been increasing over time. When we're looking 30 years down the line, we're going to see, maybe 10 million people a year dying from antibiotic resistant organisms, and antibiotics just aren't keeping up. So what we've been moving towards is the idea of using some of these natural viruses that inhabit your body that already attack these bacteria to be able to eradicate these bacteria rather than using antibiotics. So that's something where I would love to see a lot more attention to research, because most people don't even know that we've got all these viruses that are capable of killing bacteria in our bodies. And one of the things that we really are doing strongly now is just taking microbiome samples and hunting for those viruses that kill the bacteria and making them better viruses so that they're just better at what they do, so that we can use them as antibiotic alternatives.
Great, and we have time for one more brief question. So I'd like to hear your thoughts on how do we increase public awareness about microbiome research, whether in public health issues or how we communicate microbiome research, or that could also, you could also talk about how do we increase awareness of individuals who might be interested in getting into microbiome research or in training. What are your thoughts on how we can continue to make sure we recruit the next generation of microbiome researchers?
Well, in terms of just gaining public interest and understanding, obviously, and this is a problem that's been happening for years, but improving science communication amongst the public, essentially, and that really relies on scientists, trained scientists, to become journalists or reporters in science, not just relying on journalists who don't have scientific training. I think that's really important. I think a lot of people in graduate school in particular, some realize they don't want to go into academia, but there's a whole, host of different career paths that you can take after you get your doctorate degree, and so these are opportunities, I think, where we can really improve those gaps in science communication. And then, getting out in the community and taking your work and, holding workshops or seminars that are community focused. So not just talking to your colleagues in a seminar, but hosting cafes. At UC Davis, we have a science cafe, and scientists will go and host a talk at a local coffee shop, and that the public is invited to come to those. So more events like that, and just a lot more science communication from real scientists in lay language.
Very good. Anyone else have thoughts?
I'll go. All right. I think as far as public awareness, I'm not sure where my mom read it, but as soon as she realized that the microbiota can govern someone's weight gain, it became something that she wanted to talk about to friends and family all the time, and became much more interested in what I did. So I think there is a public perception that the microbiota is governing things as far as host health and fitness. So but sometimes I think there should be much more caution, because there's a lot more, I think, misinformation around what we know about the microbiome that can feed a lot of the probiotics and a lot of the idea that, things outside of diet right now can regulate your microbiome composition, that can regulate your own, athletic ability or weight gain or anything of that nature. But as far as getting people interested in microbiome studies, I think as the field grows, people become much more interested in it. And I think by default, it's becoming one of those things where it was kind of a black hole. You had a whole series of uncultural bacteria that were doing things, that we're beginning to learn more mechanistically what they do. And I think that it's one of the fields that invites a broad array of skill sets. I mean, you can be a bioinformaticist and study the microbiome, and then you can be an immunologist who studies the microbiome. You don't necessarily have to be a microbiologist who studies the microbiome. And I think that also has opened the door to a lot of people that might not have directly been interested in the field that have found themselves answering some of these very broad questions.
And real quick, I know we're running out of time, but I just wanted to make a point. What Dr. Brooks said also tied to something that Dr. Guthrie said earlier, which is, don't just base conclusions off of 20 people in a study that aren't diverse. And so I think that ties into rigor and reproducibility and making sure that our conclusions aren't, these just one conclusion fits all type of studies. And I think that'll help also improve what we communicate to the public and not going back and forth and losing trust basically from the public with scientists.
Great points. So that brings us to 6:01. So I want to wrap up now. If Dr. Pride or Dr. Guthrie, if you had burning comments, I want to make sure you get those in.
No, my only burning comments are really just thank you for getting this together, and I hope everyone was able to enjoy this or at least learn something or take something from this that they can potentially use in their lives or in their education, et cetera. I think it's great that you guys organized this for everyone.
Great. So as the co-lead organizer for Black and Micro, I want to say thank you to all of our fabulous organizers on the organization team for your work in making this week possible. This is our last panel of the week, but certainly not the least. So I just want to reiterate my gratitude for all of the work that went into making this possible. I also want to thank our sponsors again, and of course, thank you to all of our attendees who registered, who watch in real time, who watch on YouTube later. We've really gotten a lot of great engagement from all over the world. So we're all just thrilled that we've had like such a great time this week. But we do have one more day of Black and Micro Week tomorrow. We're going to spend the day reflecting on the past week, talking about the highlights and the lessons that we learned, the things that we found interesting. We're also going to provide a platform for our community to tell their stories about the challenges and barriers they've faced in their pursuit of microbiology, especially as Black scientists. As Dr. Montgomery discussed on Monday, we believe that truth-telling is important. It's a place where we, along with our allies, can come together to rebuild the oppressive systems and structures that have stolen so much discovery and innovation from the world historically. So we hope that you'll join us on our Twitter and Instagram platforms for these discussions. And just thank you again to our fabulous panelists for your time and your expertise and your insight. And we had a great conversation. So thank you all.